or osseous tissue, another specialized type of connective tissue. The first one that we have tackled was on cartilage. The bone or the osseous tissue is a rigid form of connective tissue with calcified extracellular components or ICS. So it means that it has a solid matrix. So the solid matrix deviates from the ICS of the connective tissue proper in the body, which is gel-like in nature. The bone tissue or osseous tissue is composed of the following: bone cells, collagen fibers, and a solid matrix. So there are three components of the bone tissue. Of the bone tissue. Let's go first to the general characteristics of bone tissue. It is solid, hard, and brittle ground substance which is apparently homogeneous, abundant osteocytes with cytoplasmic processes, and highly vascular. Take note of these three general characteristics of the bone tissue. The bone tissues have six important functions. Number one, internal support of the body. Just imagine the body without the bones, so the body cannot stand up, and other structures of the body will not be supported. Number two, attachment of muscles and tendons essential for locomotion. And we know here that the muscles are involved in contraction. So they are the ones responsible for locomotion. But since most of the muscles are attached to bones, so the bones and muscles and tendons are involved in body movement. Number three, protection of vital organs of the body. So we have one, no? The orbital bones protects the eyes. The rib cage protects the lungs and the heart. Then we have the pelvic bones protects the female reproductive system. And number four, the bone tissue or the skeletal system is the storage site of calcium and other inorganic substances of the body. That's why they are involved in the regulation of the homeostasis of the body. And number five, we have site for blood forming tissue. So we have the bone marrow wherein it houses all stages of bone cells. And of course, the homeostasis, the regulation of the internal environment of the body. Homeostasis. Let's go into the chemical composition of the bone matrix or the ICS. So it is made up of organic matter, or we call that organic matrix, wherein 95% is collagen and the amorphous ground substance is only 5%. So we know that the amorphous ground substance is the ICS and chemically made up of glycosaminoglycans or GAGs with protein. Okay, so the glycosaminoglycans in bone tissues, they have chondroitin sulfate, keratin sulfates, and hyaluronic acid, the same as in the connective tissue proper. However, the glycoproteins present are sialoprotein and osteocalcin, both strongly bind calcium. Then collagen fibers, the same as in the connective tissue proper. Type 1 or type 2 collagen fibers may be present. So these collagen fibers, if we will bring this to the laboratory, they are resistant to dilute acid and relatively insoluble to solvents that are used to extract other collagen types. Then we have 50% of the dry weight of bone matrix are inorganic salts. The biggest concentration will be calcium and phosphorus. So if the two combines, a chemical compound is formed. We have the hydroxyapatite crystals, and we have the formula below. Then aside from the two, we have bicarbonates, we have citrate, magnesium, potassium, and sodium. So all of these inorganic salts are the minerals of the body, and they contribute to internal homeostasis. The bone cells in the body, they are made up of actually one. No? We have the precursor of all bone cells. We have the osteogenitor cells, and this can be transformed to osteoblasts or osteoclasts. The osteoblast will later on mature to become the osteocyte. So these four types of bone cells originates from the precursor cells we call osteoprogenitor cells. Osteoprogenitor cells, if they combine, they form the osteoclast. So we have here osteocytes, osteoblasts, and we have the osteoprogenitor cells having the osteogenic, we call sometimes refer to them as osteogenic cells. They have the capacity to form osteoblasts or unite to form the osteoclasts. We have the precursor of bone cells. We have the osteoprogenitor cells, sometimes referred to as stem cells. These osteoprogenitor cells, precursor cells, or stem cells have osteogenic property, or that's why we refer to them as osteogenic cells. So they have the capacity to become an osteoblast or join together to form osteoclasts. Now, the characteristics of the osteoprogenitor cells, they have a pale staining oval or elongated nuclei, and if you will stain them, they will give us a color between a pinkish appearance or bluish appearance, no, of the cytoplasm. So we have an inconspicuous acidophilic or faintly basophilic cytoplasm. So these are the youngest cells, the precursor cells, or stem cells. Basophilic cytoplasm. The osteoprogenitor cells are found on the free bony surfaces in the endosteum, in the periosteum, lining the Haversian canals, and we have the epiphyseal plate of growing bones, especially in long bones. So, because of the osteogenic property, they undergo cellular division or transform into an osteoblast or unite to form osteoclast. Take note of the from the osteoprogenitor cells, the cells will form the youngest bone cell. We have the osteoblast. So they are the young bone forming cells. And their main function is to form the solid matrix. And usually they are found on the surface of developing bone, especially in long bones. They are found in the epiphyseal part of long bone. And the appearance of the osteoblast, they will appear to be an epithelioid layer of cuboidal or low columnar cells connected to each other by slender, short processes. Now remember, during the formation of the connective tissue, we have the undifferentiated mesenchymal cells, and these cells have cytoplasmic extension. Now going to the formation of bone in the body, these undifferentiated mesenchymal cells will have still the short processes, the cytoplasmic extension. They will be carried in the formation of the bone cells, especially if the bone cells are still in the young stage. Now we have the osteoblasts with slender short processes. The nucleus of the osteoblast are found at the periphery or are often found at the end of the cells farthest from the bony surface. The difference between the osteoblast and the osteoprogenitor cells will be the color of the cytoplasm under the microscope. The osteoblast will have an intensely basophilic appearance, so deeply blue in color, due to its large content of ribonucleoprotein with well-developed Golgi complex. From osteoblast, these cells will be developed to become a mature bone cells we call the osteocytes. So these are the principal cells of fully formed bone in the body. Just like the cartilage, the osteocytes are enclosed in lacuna, in a cavity or in a lacuna with the calcified ground substance. And the only difference between cartilage and bone tissue is that a cartilage tissue, one cavity or one lacuna may contain 2 or 10, maximum 10 chondrocytes, while the bone tissue, the ratio is only 1:1. One lacuna or one cavity, one bone cell. And the shape of the osteocytes, usually they will be flattened or they will follow the cavity wherein they are found. The osteocytes have slender, numerous processes extending into the canaliculi in the surrounding matrix. We'll go into this when we reach the cross-section of the bone. Now, comparing to the osteoblast, the cytoplasm of the osteocyte are less blue in color because they have less affinity to basic dye, and the jewel bodies are less conspicuous. That's why they are lesser blue in color compared to the osteoblast. The osteocytes, they play an active role in the release of calcium from bone to blood. That's why they are involved in the regulation of the homeostasis of the body. They participate in the homeostatic regulation of calcium concentration in body fluid. From the osteoprogenitor cells, if the different osteogenic cells will form and unite, they will become a giant cell we call osteoclasts. So the osteoclasts are the bone-destroying cells of the body, and they are described to be giant cells that may contain as many as 50 nuclei that appears foamy. No? So the cytoplasm appears foamy and have a ruffled border, particularly at the surface of the bone. So their main function is bone resorption. And they are usually found in shallow concavities of the bone surface, referred to as house shaped lacuna. So you can see in the illustration the ruffled borders of the osteoclast, the nuclei inside the cell. It may contain as many as 50 nuclei. These are because the osteogenic cells have combined together, contributing to the number of nuclei of the cell. So we have here illustration of the osteoclast found in concavities of the bone tissue. Okay, another illustration wherein we can see the ruffled border, the nuclei inside the osteoclast, wherein they appear as hollow because of the stain that was used. So we have here histoenzymologic coloration of the tartrate-resistant acid phosphatase. So they appear as giants. Okay, so this is a summary of the different formation of the bone cells in the body. Okay, we start again. We start first with the osteogenic cell. They could be formed into an osteoblast or young bone cell or unite to form a giant cell. So we have here an illustration of an osteoclast in a cavity with the ruffled border, foamy cytoplasm, and different nuclei inside the cell. Again, we have the summary of bone cell development from osteogenic cells. These are the osteoprogenitor cells. They will be transformed into osteoblasts. So they will, of course, undergo first mitosis or cellular division to form osteoblasts, then mature to become an osteocyte. I would like here for you to focus on the illustration of the osteocytes. So they have this cytoplasmic extension, because we will understand better how are the canaliculi are formed in bones. Then from osteogenic cells, the cells, if ever they will unite, they will form into a giant cells we call osteoclasts. In bone remodeling, there are 6 stages. We have the resting, activation, the resorption, reversal, formation, and mineralization. So in the resting stage, there will be no bone resorption. No? So the bones will be transformed from a young cell to adult cells, so mature bone cells from osteoblast to osteocyte. Now, if there is an extra bone cells or there is an overpopulation of bone cells that are formed, the osteogenic cells will be activated to become osteoclasts. So if the osteoclasts are formed, they will actually undergo bone resorption to balance the population of bone cells that are formed. So we have activation to resorption. After resorption, the osteoclast will revert back to the osteoblast formation. So there is reversal, and the osteoblast will be formed to become young bone cells, and there are bone deposition. The osteoblast will synthesize the matrix and a bone tissue is formed. So we have from mineralization again to resting stage. So take note of the 6 stages: resting, activation, resorption, reversal, formation, and mineralization. So just also take note from the osteogenic cells. The osteogenic cells will be formed into an osteoblast. The osteoblast will become mature to become osteocyte, while some of the osteogenic cells will unite to form giant cells. We have the osteoclast, no? And they will undergo bone resorption. Now, this will happen during the time that there is ossification of bone cells in the body. So any extra bone cells, no, that are formed will be resorbed by the osteoclast to balance the population in the body. And we have this process of apoptosis wherein the cell will just undergo destruction, self-destruction, or cell suicide. Now, this is a normal process that the cell will undergo to prevent overpopulation of bone cells in the body. We go now on the types of osseous tissue in the body. So one criterion that classify the osseous tissue in the body is based on macroscopic morphology, or how they appear, no, using our eyes. So we have the first one, we have the cancellous bone or spongy bone or substantia spongiosa, or commonly referred to as soft bone. Then on going to the outer part of the tissue, we have the compact bone or substantia compacta, or commonly referred to as hard bone. Now, just imagine a chicken leg bone. Now, if we open up the leg bone, usually we can see in the inner part of the bone, we have the spongy bone or the soft bone, and on the outer part of the bone, we have the substantia compacta or compact bone or hard bone. Let's go first or describe the spongy bone. So the spongy bone is simple and less organized architecture. So what we can see here will be just a network or anastomosing bone trabeculae or spicules. So this network arrangement of the soft bone contains cavities with endosteum containing bone marrow. Take note, in the bone tissue or spongy bone, there is an absence of Haversian system. Or we have another term for Haversian system, the osteon. On the uppermost portion of the illustration, you can see just towards the hard part of the bone, a small tissue is taken out and can be observed under the microscope. We have the concentric arrangement of the osteoblast and the osteocyte. But this is not yet the Haversian system. Okay? In this illustration, we still have the osteoclast, and what is not shown here is the Haversian canal at the center. So take note, absence of Haversian system or absence of osteon. The compact bone have an organized architecture. They appear more solid with fewer cavities. So under the microscope, we could see the regular arrangement of the concentric layer of the bone tissue, or we call that circumferential lamellae arrangement or concentric lamellae arrangement. So this forms what we call the Haversian system or the osteon. So take note, in compact bone, presence of Haversian system or presence of an osteon. We have here an illustration of a compact bone. So this is found towards the outer part of the bone tissue, and it is composed of concentric arrangement or layer we call the Haversian system. The outer membrane that supports the inner part of the hard bone or compact bone is what we call the periosteum, made up of fibrous layer and cellular layer. I would like you to focus on one concentric arrangement we call the Haversian system. So at the center, we have the central canal or Haversian canal, and surrounding the canal is a concentric arrangement of the bone tissue. We call each concentric layer a lamella or a concentric arrangement. And within this lamella, at regular intervals, are the cavities, and within the cavity, we have the osteocyte. And I would like also to focus your attention on the smaller tubular arrangement from the lamella towards another lamella. We have the tubular network arrangement, and these are the canaliculi. So if you will be asked what composes an osteon or Haversian system, we have the central canal or Haversian canal, we have the concentric layer or lamella, wherein the cavities are located with osteocytes, and we have the canaliculi. Now, in the central canal or Haversian canal, it is composed of blood vessels, both the arteries and the veins, and of course, the lymphatic vessel is composed also in these areas. And the main purpose of the vessels, especially the blood vessels, is to give nourishment to the bone cells. To the bone cells. So the arteries, especially the arteries, will supply blood to the bone cells of the osteon. In this illustration, we have both the cross-section and longitudinal section of bone tissue. So for the cross-section, you focus yourselves on the surface part of the bone. We have the different Haversian system or the osteon on the surface, which is also protected by a membrane on the outside. We have the periosteum. Now, in longitudinal section, we can actually observe the canals that traverses the bone tissue. Going upward, we have the Haversian canal, and on the sideward, transverse section, we have the Volkmann's canal. And each of these canal will be composed of blood vessels to give nourishment to the bone tissue. We have here another illustration of the cross-section and longitudinal section of a bone tissue. So on the surface, we have the different Haversian system. Now I would like you to focus on the Haversian system that give us the illustration of the concentric arrangement of the lamella of the bone tissue or the lamella, and at regular interval we have a cavity, and within the cavity we have the osteocyte. I would like you to focus on the illustration of the osteocyte with the cytoplasmic extension. So during the time that the bone will undergo deposition of the matrix, the cytoplasmic extension will be covered with the solid matrix, and later on, the osteocyte will withdraw its processes. So what will happen to the processes structure? It will become the canaliculi, so giving us again the smallest tubular arrangement within a bone tissue. So if you will be asked what are the different tubules that are found in a bone tissue, we have the Haversian canal, we have the Volkmann's canal, and we have the canaliculi. So these three tubular structures function to supply blood to the osteocytes within the solid bone matrix. So, three tubular structures that give nourishment to bone cells. We have the Haversian canal, the Volkmann's canal, and the tubular structure we call the canaliculi. In this illustration, we have the longitudinal section of a bone tissue. So again, we have canals going upward into the Haversian system, the Haversian canal, and traversing the bone to the sideways, we have the Volkmann's canal. As mentioned, again, these canals provide nourishment to the bone cells of the tissue. Okay, so in this illustration, we have the cross-section, and we have here several concentric arrangement of the tissue we call the osteon. So how many here? 1, 2, 3, 4, 1, 2, 3, 4, 5, 6. No, the sixth one on the lower side, we can only see the part of the Haversian canal. So each concentric arrangement, several of them, we can count 1, 2, 3, 4. Maybe an osteon may have 4 to 5 concentric layer or lamellae. So these are the morphological characteristics of the bone, very easy to identify under the microscope because of the arrangement of the osteon or Haversian system. We have here again the different osteon bone tissue with the parts labeled. Okay, so we have on the upper part that is circled by an orange color, we have the Haversian canal, canaliculi, lacunae, and the lamellae. So these are the Haversian system that are found in compact bone. So this is only a 10x or 10 magnification under the microscope. So here we have a larger illustration of the bone osteon. Okay, so this is 40 magnification or 40x. So again, we have here a big canal, the Haversian canal with its concentric arrangement. We have the lacuna within the bone tissue, and inside the lacuna, we have the osteocyte with the canaliculi that interconnects the different lacuna. So here we have a detailed description of the Haversian canal. So at the uppermost, we have the Haversian canal with the concentric layer, the lamellae, and within the lamellae, we have the lacuna, and of course, the membrane which cement together the osteon, we have the periosteum, and on the lower portion, we have the interconnection of the osteocytes within the lacuna. So they have an interconnection through the cellular processes, which will later on will be withdrawn by the osteocyte, mature bone cells, and these processes will become the canaliculi. We have here again an illustration of the interconnection between osteocytes within the lacuna through the cellular processes, which will later on will become the canaliculi. Let's go now into the membranes that protects the bone tissue. On the compact bone, we have the periosteum. This is a thick membrane made up of connective tissue again subdivided into the fibrous layers composed of connective tissue fibers. We have the collagen fibers, and we have the lower layer, the cellular layer, which has the osteogenic cells. Okay, so we call this as the osteogenic layer. And going to the soft bone or spongy bone, we have the endosteum, which is also made up of connective tissue. And compared to the periosteum, the endosteum is a thin, vascular, connective tissue membrane. The periosteum contains collagen fibers, and these collagen fibers has been identified to be type 1 or type 2 collagen fibers, and these fibers, the name of these fibers is credited to William Sharpe, a Scottish physiologist and histologist. And since these Sharpe's fibers traverses the periosteum going to the endosteum, it is also referred to as perforating fibers. The periosteum has several functions. One, attachment sites for the muscle, tendons, and ligaments. Two, nourishment of the bones and muscle. Three, bone sensitivity. So in here we have the nerve fibers, innervation of the periosteum, and also involved in bone growth and repair, and serve as a periosteal hinge. The endosteum have three important functions: the appositional bone growth and development, bone repair, and bone remodeling. For bone repair and bone remodeling, it's because of the osteogenic cells found in this layer. So they are involved during traumatic injury or during bone fracture or bone resorption to have a renewed type of bone cells. Okay, so we have here a summary of the bone membranes. We have the periosteum and the endosteum. So if we go into the periosteum, we can see the different structures that are involved here, and into the endosteum, the different structure also observed here during microscopic observation. So we have here on the lower portion the components of the periosteum and the endosteum. We go now into bone growth and development, which we call the ossification process. There are two types of ossification process involved in the body. We have the intramembranous ossification and endochondral ossification. Now, for intramembranous ossification, this will involve the flat bones of the body, and usually this is the grow in width of bones. So in this illustration, we have the cranial bones wherein made up of flat bones. So during the development of intramembranous ossification, the undifferentiated mesenchymal cells, of course, are involved, and they will conglomerate to form what we call the primary bone plates. And later on, the thin bone of the body or the flat bones of the body are formed. So this is in flat bones of the body, especially in the cranial bone. Another illustration of intramembranous ossification from the undifferentiated mesenchymal cells to conglomeration of the mesenchymal cells to form the primary bone tissue, forming into a thin bone or flat bone. So in this illustration, we have now a developed thin bones of the body. So especially in the cranial bones, no? The cranial bones will grow or develop towards each other, which will later on will the edges of both flat bones will become now sutures, which will later on harden to become part of the cranial bones. So in this intramembranous ossification, there is grow in width of the bone. So we have now here the process of endochondral ossification involving long bones of the body. So we have both ends of a long bone. We have the metaphysis on both ends and the epiphysis. Now we know that the epiphysis is the growth plate of long bone. So during the time that a long bone is being developed, we have the mesenchymal cells that are involved here. From the mesenchyme tissue, they will be formed into a hyaline cartilage. That's why the endochondral, the model is a hyaline cartilage model. And later on the diaphysis of long bone, we have a bone collar formation. And this bone collar will actually be provided with vessels. And here, the start of ossification will begin. So we have the primary ossification center at the diaphysis part of long bone. So in the primary ossification, it will be actually be supplied with blood vessels. So there is actually growth of bone going upward. So the growth will be towards upward and towards also the lower portion, so there is bone growth in length. And from the primary ossification center, both edges of the long bone will be formed into secondary ossification center at the epiphysis part of long bone. So take note, the primary ossification center will be at the diaphysis and secondary ossification center will be at both sides of long bones in the epiphyseal plate. So secondary ossification center. So the bone will now be developed slowly, and in the diaphysis there will be different zones that are formed. We have here the bone collar formation at the diaphysis part of long bone. So you can see here the different cells that are involved from hyaline cartilage to bone tissue. So here again the perichondrium in hyaline cartilage are involved because of the osteogenic property of the cells here. We have the young cartilage cells, which will later on form the cartilage model of endochondral ossification. So in this illustration, we have the diaphysis wherein the primary center of ossification is involved, and we have in the epiphysis we have the secondary center of ossification that is involved. So in between the two we have the growth plate. So in the growth plate, it is here where the different zones of the cells are formed, or different changes of the cells from cartilage to bone cells are involved. So in this illustration, we have now the different zones that are formed during endochondral ossification in the epiphyseal plate of long bones. So we can see here different zones: the resting zone proliferating zone, hypertrophic zone, and ossification zone. Then later on, the spongy bone are formed in the body. So we have the trabecular bone. Now in the resting zone, there will be from the cartilage formation of the cells, there will be no changes of the cells. So from young cells until the cells will grow in number, we have the proliferating zone, and some of the cells will grow in size. Cartilage cells will grow in size to form the hypertrophic zone, and later on, these large cells now will become bone cells. Slowly will be transformed to bone cells. We have the ossification zone and formation of the spongy bones in the body. This is now the last slide of our lecture.